All right, hi everybody, and thank you for attending our online seminar. Today we have a very special guest speaker, Daniella Goldfarb. Dr. Goldfarb is from the Weizmann Institute, and probably many of you guys know she has pretty much paved the framework for many things what we consider nowadays as common practice. She laid the framework for pretty much using gadolinium for deer distances. She has done technology advances, and I don't know, like pretty much has been just a centerpiece of the EPR community for a long, long time. And therefore, we're very excited to have her here. And especially, I want to give her a shout out. I mean, please understand, look, she's right now in Israel, Weizmann. She's so dedicated to EPR that here she is giving a talk. And I really appreciate that. So for today, she's going to talk about the ins and outs of the gadolinium fluorine ENDOR. And I'm actually very excited because, of course, this is like the fluorine distances and the whole determination is like the next big thing. And so I'm looking forward for Daniella's talk. And with that being said, Daniela, I give you the floor. Thank you.
Okay, thank you very much. And thank you for inviting me. I'm really sorry that I cannot see you and talk to you directly. I have to look at the screen and imagine that you are there. So what I want to tell you is about what we've been working, I guess, in the last 5 years or so, and this is fluorine ENDOR with gadolinium spin labels, and how do you say, the ins and the outs of this. So let's get started. So when we think about structural information from magnetic resonance, usually we can either measure interactions between different nuclei. Yeah? So this could be within the same nuclei or different nuclei. And this usually is done by NMR spectroscopy. We can also get information through interactions between electron spins, again, same type or different types. And we can also get information from electronuclear interaction, and this will be the subject of this particular talk. So let's start with the structural information we get from dipolar interaction between two electron spins. And this is pulse dipolar EPR that has been very popular in the last, I don't know, maybe already 15, 20 years. And this is one of the sequences. It's the double electron-electron resonance sequence. And with this, we can measure electron distances between 16 and 80 and angstrom. And if you deuterate your protein, then it can go even longer. Now, and the dipolar interaction between two electron spins, here is, you see the equation that I guess you are all familiar with, and it depends on the orientation relative to the magnetic field. So here, these are the two spins. There's a vector connecting the two, and this is the angle this vector makes with the field, and this distance. So normally, if we work in frozen solution, all theta angles are possible. We know how to deal with it, and you can extract the distance. And this has been really very, very useful, and I think it gave EPR a lot of publicity, and in the last, I don't know, 15 years. So if you want to work with proteins, usually they are not paramagnetic, so where are your spins? So you use spin labels, and you see this is a pretty old paper from 15 years ago, and here is the protein. You put your labels. These are nitroxide, and this is the MTSSS spin label that has been around many, many years, so it's still old but kicking, right? It's not that it's dying, it's just to say that it's been around for a while. And you can also use gadolinium spin labels, and also these are not babies anymore, maybe they are teenagers, and there are still trityl and copper and different type of nitroxide and manganese. But in this work, I will concentrate mostly on gadolinium spin label, maybe a little bit on nitroxide. So there is pulse dipolar spectroscopy is wonderful, but there are some limitations. So what are they? First, the distance to measure below 20 angstrom is not very easy. And now when you measure short distances and you have two spin labels, so this the spin label takes quite a bit of they contribute to the distance, and then your error becomes larger. Also, spin labels are rather large, so you usually you label it at exposed sites, but if you have, let's say, dense sites within the protein, then you cannot put a big label there without disturbing the structure. And if you want to look at small ligands that interact with the protein, then you have a small ligand and a pretty big spin label. So this is also not so great. And finally, if you want to measure in excess of the spin label, and you use the same type of spin label, this is very difficult because you have this huge background. So there's place for different method. And here you can add electron nuclear distance measurement. So this distance, of course, do not come to replace the Deer type experiments, but they are complementary. Yeah, they have their advantage for certain problems. So how do we measure distances by ENDOR? So this is the equation for electron-electron dipolar interaction. So we saw it before, the distance, the angle, and here is just the Bohr magneton of the electron. So now if we measure with nuclei, so we have an electron and a nucleus. So we have this term that is different, and then this electron nuclear frequency, of course, will be smaller. This is still the same. So let's look what are the nucleus that are popular that we can use, and what type of hyperfine coupling they will give for 1 nanometer distance. So protons, it's great, right? Because it has the largest gamma, the gyromagnetic ratio. So for 1 nanometer, you can get splitting of 79 kilohertz. That usually is easy to resolve. The problem with protons, you have too many of them. So it's not really very specific in a system. You go to deuterium, then this really drops, so it's not very useful. C13 also not that useful. Phosphorus is already better, and fluorine is really very similar to proton. So actually, with for distance measurement, phosphorus was already used, and this is a paper from 2005. It was a collaboration of myself and Gunnar Jeschke. And in this case, we could measure up to 10 angstroms. So of course, if you will go to fluorine, you will extend this range. So just to give you some introduction about ENDOR. So you have 1 electron coupled to 1 nucleus. So you have 4 energy level diagram. Here is an EPR transition. And there's another EPR transition here. And we have 2 NMR transitions, ni alpha, ni beta. And what we measure in ENDOR is really these NMR frequencies. So if we would do an NMR experiment or an ENDOR, then we have these 2, the splitting. This is ni alpha, this is ni beta. They are centered around the Larmor frequency, and the splitting is the hyperfine coupling. This applies for the weak coupling case, and as we will be talking only about W band measurement, 95 gigahertz, so we are always in this regime. Not always, but at least for what we measure. So how do we measure? We use the MIMS ENDOR experiment that has been, I think it's from the 60s, very, very long time ago. So this is the sequence. You actually collect a stimulated echo, and during this time here, you apply a pi pulse and you measure the echo as a function of the frequency of this pulse. In our lab, in order to gain SNR, we apply a CP detection sequence. So we we apply this series of pi pulses, so we get many echoes and we add them together. So this is just for SNR application. And here is just an example. So if you don't have your RF on and you look at the sum of all these echoes, you just see a flat line. But if you apply the RF, then at a certain frequency, you see this powder pattern. This is the perpendicular, this is the A parallel, and this is the fluorine Larmor frequency here in the middle. But you know, things are never great. So when you do MEMS ENDOR, it has two bugs, but actually one big bug. So the ENDOR effect, this is the intensity of these lines, they are relative to the echo, depends not only on the hyperfine coupling, but also on this time tau. Yeah, here, the spacings between the pulses, and the you get, so you see that if you depends on your tau, you will get a different ENDOR effect, and for certain taus, you really get blind spots. There's no ENDOR effect, and if you want to maximize your ENDOR effect, you have to choose your tau this way. So if you have very, very small hyperfine couplings, if you want, and this happens for long distances, then your tau has to be very long. And then what happens, like everything, the phase relaxation hits you in the face. Yeah, so if you want to measure long distances, you need to apply long taus, and in this case, you are limited by the what we call the phase memory time. Okay, so we have to remember these blind spots. Okay, so now this, so as I said, this is the, and we consider only dipolar interaction because we want to look at long distances. So here's the equation. We can have all type of, it's a frozen solution. So we have all possible thetas. And here is the ENDOR powder pattern without the blind spots. And here is the perpendicular one, and here is the parallel. So now what happens when you have a nitroxide, for instance? So this is just a calculated spectrum, and you have a powder pattern. So, and your excitation is limited, and you cannot excite the full powder pattern. So you excite only a small bit. So in this case, you don't excite all thetas, and you get what you call orientation selection. So if you will measure here, you know, you will have different set of thetas if you will measure here, and this will give you different spectra, and it means that you need to collect all of them if you want to get the right distance. So before I go to show you all kinds of examples, so then, you know, why fluorine? I already hinted to this before. So first, you get high sensitivity. As I said, the gyromagnetic ratio is very similar to proton. So this means high sensitivity, large couplings, and you can reach relatively long range. But what is also important, it has a low abundance in biological systems. So you can really put your fluorine. So it's like you have one label that is your electron spin label that is really big, but then you have a tiny atom in a molecule that gives you your nuclear spin, and fluor is not very abundant. So then that's how you introduce your specificity. And this is really the first work that really started this whole thing of fluorine ENDOR. This was a work by Marina Benatti in 2019, where she showed really very nicely with the nitroxide. Here are the models that she used. And here is the nitroxide spectrum at W band, and she measured here, she and her students. And you see that you have a different spectrum. Yeah, this is mostly the parallel one. And then when you add them all up, you get the sum, which is more or less the powder pattern. And here she shows that she can measure up to 15 angstrom and here 12 angstrom. But she needs to do all these measurements, right? So she has to do all these measurements. And then by doing this analysis, you can get not only the distance, but also the orientation of the spin label in the molecule. And this can be very important information if this is what you are looking for. So now, okay, so nitroxide really works. So for why gadolinium? Why bother with gadolinium? And I hope that you will see this later throughout the talk, why bother? So let's let's look at the specific EPR properties of gadolinium. It has a spin 7.5, and so here, if you work at 95 gigahertz at relatively low temperatures, so then the populations of the different EPR levels are different, just according to Weizmann. And the spectrum, if you 10K, let's say, is this gray curve. So it's this central transition, this red one, superimposed on all these other transitions. The central transition does not depend on the zero field splitting, and therefore to first order, it depends to second order. So if the ZFS is small and you work at high field, this is a very narrow line, and it's very intense, and it doesn't have orientation selection. And then you see here the spectrum that is measured at different temperatures. So you see, if you look at 14K, this is this one. So you see the central transition is very strong. So if you work here, it will really be like a spin half. But then if you go down in temperature, all these other transitions start to contribute, and this one is getting smaller, and this has some advantages, and I will talk about it later. Okay, so now the ENDOR, right? So now the energy level. So if we just look at the central transition, then it's really like S-half-like. Yeah, so you have these two levels of the electron, and they are split by the nucleus, and you have these two EPR, these two NMR transitions, and their frequency is given by Larmor frequency minus the N sub S. In this case, it will be either minus half or plus half, and the hyperfine coupling, and you get these two lines that are centered around the Larmor frequency, just like I showed you before, and there's no orientation selection. And this was pointed out very nicely by Nick Cox and his students, that showed that you can do this fluorine ENDOR with gadolinium without orientation selection. Okay, so let's start by comparing different types of spin label. So we have here, and this is work that was done in collaboration with Angela and Venkai, and that made all the protein with the fluorine, and Yang Ping, who gave us the trityl, and Su Cheng that we work with and gave us the gadolinium. So we have here several spin labels. Here is the nitroxide, the usual one. So you stick it here, and you have here your fluorine. In this case, it's 3 fluorine. This is for ubiquitin. For GB1, there is 1 only here. Yeah, so it's either you have this amino acid, or you have this tryptophan that has only 1. So these are the 2 different types of nitroxide. This is gadolinium, and this is the trityl. So each one of them has a tether that has a different length. So in principle, we are just interested in the distance between the, let's say, the beta carbons. But the minute I add the spin label, if it has a long tether, I'm reducing my sensitivity for the distance, because let's say I can measure only up to 15, and if the tether is large, then I cannot really access long distances. So let's just compare these guys and see how they perform. So first we have to look at the spectrum, right? When we think about sensitivity, the EPR signal is important. So this is all on the same scale. This is the nitroxide, this is the trityl, and this is the gadolinium. So in principle, the trityl should have the best sensitivity because it has the narrowest line. But now we have to worry about the intensity of the EPR line. We have to worry also about how fast we can accumulate. So if you have short T1s, you can accumulate faster. And then about the decay. So here these three are compared, and you see that the trityl, what was really pretty disappointing is that it really decays pretty fast. We hope that it would decay slower. Anyway, you see gadolinium is not doing too bad. So we get good sensitivity. We have a strong signal with fast repetition rate, and we have decent phase memory time. And as I said, we don't have orientation selection. And most of the work on this ENDOR with fluorine was done by Alexei Bogdanov, who was a young co-worker in my group. Okay, so now these three were put in the same place. And then we also looked for this copper double histidine that was not put in the same place, but we were interested to see whether we get a better resolution. Like it's a rigid spin label, so we shouldn't get a large distance distribution. So you see the nitroxide has the shortest distance because it has the shorter tether, gadolinium a bit larger, and trityl is the longest. So you almost, for this distance, you're almost close to your maximum resolution. And for the nitroxide, we had to measure all these different orientations. So this is really 4 spectra, and this is their sum. And indeed, you see that you should get nitroxide has a shorter distance than gadolinium than trityl. So here, just to see this, so you see that for ubiquitin in this position, you see the nitroxide and the gadolinium give more or less the same distance, but you don't resolve the trityl. And here you get for the nitroxide, you resolve. For gadolinium, not really. They have these shoulders. And for the trityl, it's really very difficult. So I think that trityl is maybe not the best spin label for this. Okay, so we saw that you can do these measurements with gadolinium. It has certain advantages. And then the next thing you think of is, okay, so we want to go in cell. And this is what we did. So here are these 2 proteins, the ubiquitin and the GB1. And we took 2 different mutants for each. The fluorine was in the same place. And this is the collaboration with Angela and Sucheng that makes this spin label for us. And so these are just the ENDORs that we measured. And if you analyze them with a single distance, the fit is sort of okay. These are the distance that you get. You see at 1.5, we really have no resolution, just these shoulders. Now, if you put a Gaussian distribution, you see it fits a bit better. And now Angela did these measurements also with PREs, NMR PREs, when you look at the effect of the gadolinium spin label on the fluorine relaxation. And you see that this is the distance that one gets from the NMR. And then if you take this number, you see this fit. So this is pretty good. Remember that one is done at room temperature and one is done at low temperature. So this was really encouraging. And then we went to do the in-cell measurements. So these are just usually when you do the in-cell measurement, this is your EPR So the protein were injected into the cell using electroporation. And this is the intrinsic signal. You have always manganese signal. This was done by Arina. She's an expert with this. And then this was the in-cell data. We didn't attempt to do the one that had the 15 angstrom distance. So, and the dark line is just if you smooth the data. So I show you here both the SNR. This one really had a very nice SNR relative to the others. I don't know why. And now if you compare to the solution one, then you see that you get the same distance, and this is what you expect. GB1 is a well-structured protein, so this was just a test to see that you can do in-cell measurements with this ENDOR using gadolinium fluoride. And okay, so far I showed you examples of proteins. Of course, you can do it with DNA, but you can do it also on some chemical systems. And this is host-guest complexes, and this is a collaboration with Amnon Barshir and Elad Goen from our institute. And they had this really nice system. So they had cyclodextrin, and there are two types of cyclodextrin, alpha and beta. Alpha has a 6-member ring. It's this type of sugar that you can include all kinds of molecules there. And this is the beta one that is larger. And they used fluorinated guests, so all these different types of guests, and they put it in. And now they attach to the bottom of this cyclodextrin, they attached a chelate that can take lanthanide. Okay, so this can be gadolinium or dysprosium or whatever. And they used an NMR method called CEST. And I really don't want to explain this method now. I don't have time. But so they put dysprosium in this bottom chelate, and they see that for the alpha cyclodextrin, they can see this is the fluorine around, but this is the fluorine that is in the cage, and it's shifted. And you see also for this one. But when they use the beta cyclodextrin, they see 2 peaks. So this means that they have 2 complexes. And they could tell that they have 2 complexes, but they wanted to have some structural information, why they have 2, what is their different properties. And that's when they contacted us, and we wanted to see whether with the ENDOR we can get some structural information. So this is work that was done by Manas and Alexei. And so here you can compare the alpha cyclodextrin and the beta one. So for instance, this is a really nice example because this is very weak binding. So you have a huge excess of your guest, but you see only the one that is close to the gadolinium. So this is really one of the advantages of this method. And if you analyze the spectrum, you get 95% that are the distance of 8.8 and 5% at the distance of 5.1. You get this for when you have the fluorine in the para position or when you have a CF3 in the para position. You see that the second one is very, very, very small. I think in the NMR they didn't see it. But when you go to the beta cage that is bigger, you see here 2 distances very clearly. A major one that is 75% and the other one that is already 25%. So it went up from 5 to 25, and the same goes for this guest molecule. So now we really wanted to know what, if we could get really structural information, what's the orientation in the cage. So we thought, or Amnon and Elad thought that probably this could be binding very weakly to the gadolinium, and then this would dictate the orientation of this molecule in the cage. So here So I told you that gadolinium is great because it has no orientation selection. But sometimes, surprisingly, when the zero field splitting is very large, it has. So what's nice is that you can decide where you measure, and in some cases you can have orientation selection that can help you with structure. So here, this is the central transition of the gadolinium, and you see it has shoulders here and here. So Manas decided, well, let's check and see what happens in these shoulders. And you see here, you see when you measure in this position, you see this major splitting. When you measure here, you see this powder pattern. And when you measure here, you see the inside. So this really is classical orientation selection. You can simulate this. You can see at each position of the EPR spectrum what are the orientation that are contributing. And eventually you can get the orientation of the gadolinium fluorine vector relative to the zero field splitting orientation. And you can do this with for all for all these. I don't want to. Yeah, we did it for all these, so you have different fluorines. So this gives you more information, and if, at the end, you can really get the geometry. So this, so here is the. This is, I think, the benzene ring, and you have here is the nitrogen, and we have this distance, and we have the orientation relative to the zero field splitting, and here you can see how it goes into this cage, and this is the plane of the ZFS of the ZFS tensor. So in a way, this was really exciting. And what you can do next, and I don't have time to talk about it, but if you take a molecule that you know the geometry, you know exactly where the fluorine is relative to the gadolinium, then you can determine where is the ZFS of the gadolinium. This is usually hard to calculate and very hard to determine. So this is for next year. So let's see, let's conclude. What can we tell? So from all labels, it seems that the trityl was too long and hard to predict. Copper looks very nice, but at least at 95 gigahertz, the sensitivity is low because it has a very broad spectrum, and there is some orientation selection to take care of. These are the distances that are accessible. We confirmed the distances that we measure with PREs and MR. We demonstrated in-cell ENDOR. I didn't talk about it, but modeling, when you model distances if you have a structure, you can use MMM or MTSL wizard or whatever to model the distances you get from electron-electron distance distribution. And there usually your error is around 2 to 3 angstrom, which for a distance of 40 angstrom is fine. But for ENDOR, if you measure 10, 12, 15, an error of 3 is too big. And I think here some improvement is needed. And regarding orientation selection, you can sort of turn it on and turn it off. And here is the minimum detectable concentration in the cell. Will be nice to be a bit better. So now, okay, so I hope now you're convinced, you understand, and everything is good. But can we extend the distance? Yeah, we measured up to 15. And the question is yes. So now we exploit the fact that gadolinium is high spin. Right? So before I showed you only this energy level of the half minus half that behaves just like a spin half. But if we are exciting, for instance, this an EPR transition, then we have these NMR transitions, and they will be placed somewhere else. So if I excite this 7 half, if I excite this one, right, it will show up here. and then the other one will show here. And so they will move. So let's see the picture that is easier for the powder pattern. So this is here the powder pattern, the peg pattern you get for spin for the central transition. Now the powder for the other transitions, you see, they are much broader and shifted from the center. So if you lost your resolution here, you can still measure this shift. So you can really extend your resolution. So let's see how it works. So now, unfortunately, we have these blind spots, so you don't get this nice powder pattern for each of these, but you see this, here's a blind spot, and here is another blind spot, yeah, so you see this should be, here is the classical one, and then you see the blind spots. Okay, so this is just an example for the half, and this is for the -3. Okay, so now let's introduce line broadening. And so if you introduce the line broadening and the blind spot, if you measure the half minus half, you will get this spectrum and this tiny little splitting. But if you go to these transitions here, you get, this is with the blind spot, you get this powder pattern, and you see this large coupling, and this large coupling is 7.5 A perpendicular compared to 1 A perpendicular. So you extend it by a factor of 7.5. So let's see if this works. So here is the experiment that this was done on ubiquitin in one of these proteins that I showed you, and this is measured at 2.2 K. This particular one, I want to thank Leonid and Alexander Schnegg who helped us measure this. This Alexei measured in their lab. Now I think in our lab we can measure 3K, and you see here, you barely see this splitting, but here you see it's very clear and very nice. And this is even more so for this one, you see the central transition was not resolved, but when you go to the ends, you can see and you can resolve this very, very nicely. So you can extend. So the question now is, what is what is the limit? How far can you go? So this depends on the line width. So if you have a line width of 30 kilohertz, so, and you can measure a tau that is equal to 4 microseconds, which is possible, then I guess you can go up to here, which will be, this will be 17 angstrom. If you have a lower line width, you can probably go even more. You can go to, let's say, 22, even maybe 25. And the main problem is that the ENDOR efficiency goes like 1 over to the R6. So there is a very interesting approach that has been developed by Nick Cox. And what he measures, he measured the ENDOR intensity. We measure the ENDOR splitting. So, you know, when you look at the spectrum, you see the splitting. This is easy, but you can maybe extend further by measuring the ENDOR effect, because so even if you don't have resolution, you can measure the intensity of the peak. But for this, you have to have very, very good, how do you say it? You need very, very good calibrations. And we all know that intensities are more difficult, but you can extend it further. There's just a JAX paper they published that is very nice, so you can go and see this. So we think that with this method, we can measure up to 20, push to 25. With their method, maybe 25 is more easy, but I'm not sure. But you always try what you can. Okay, so now we talk about resolution. So how about the resolution? What determines the resolution of these endospectra, right? because if we have narrow lines, we can measure longer distances. So let's see what affects the ENDOR line width. So first thing, of course, is the distribution of the distances, and so if you have a label that is not flexible, you're doing better. From our experience, this is usually between 15 and 25 kilohertz. Now you have to worry about the radio frequency pulse excitation. There's very nice work of Marina Benatti that measured all these things. So here, this is approximate the broadening from the pulse excitation is 6 to 20 kilohertz. So you should work with long pulses. You work with long pulses, and then you lose sensitivity. There is the chemical shift and isotropy of the fluorine that introduced broadening, and we've seen this in W band. And then a very important contribution is the dipolar interaction of the fluorine with either other fluorines or with dipolar interaction. So this is again in this paper. And the question is, what is the maximum resolution? And this would be determined by the coherence time, the T2 of the electron. And the question is, what is the T2 of the electron? And is it something important, or all these other things just override it? So we decided that we have to measure these in gadolinium, and this measurement actually was introduced by Peter Hofer and Mering long time ago. It's called time domain ENDOR, and these are difficult experiments, so it's not very it's not applied very often, but Alexei is really an incredible experimentalist, and he got it running very nicely. So basically what you do, it's your MIMS ENDOR, but then after you have an RF pi pulse, then you get a pi pulse and another pi pulse, and this is your echo. So you create a nuclear echo, then you transfer the magnetization back to the electron and you measure it. And you see here, so if you measure, you fix this time, and then you change this time, you see the nuclear echo. And this is done for the proton, which have much stronger signals. And then here is the echo for a tau of 10, of 20, and you see all these echoes. These are the nuclear echoes. And if you follow them, you get this decay, and this is the proton one. So now what you can do, you just fix this time, and you measure the nuclear echo intensity directly to get this. And this is done. So here we did these experiments for these model compounds. And this is a DO3A gadolinium label that we use a lot. And this is a PyMTA label that is used. So these are pretty common gadolinium labels. And here is the example of the data for gadolinium 1. And you see the echo. And of course, the SNR is much worse because you have one fluorine and not, 20 protons. And then you measure them as a function of temperature. We want to understand what determines this relaxation. And now if we plot this relaxation, so we measure them as a function of temperature, and what changes with temperature, it's the T1. The T1 had to be analyzed with a stretched exponent. So we took what is some sort of an average, and this is a measure that takes into account both the exponent and the decay coefficient. And if you plot now your 1 over T2n that you measure as a function of 1 over T1, you see that it falls more or less on a straight line, which tells you that the T2, 1 over T2, is equal to 1 over 2 T1s. And this was already predicted by John Morton in one of their papers, and this is really very nice. So this tells us that at least for gadolinium, for fluorine, for protons, things are different. I don't have time to go over this. But if you go lower in temperature, your T1 is longer. eventually you can have a longer T2N. So I think now I'm happy. You got also the resolution. And I think with this, I think with this, I showed you all the ins and outs of this gadolinium ENDOR for fluorine ENDOR. And I want to thank all the people that worked, and there were lots of them, and Alexei did most of this work, and I'm sure that you will continue to hear and see him everywhere. Manas did a lot. He started this work and then he left for a position in India. Arina helped a lot with the in cell work. Akiva was there when we started, improving the ENDOR of the instrument. And Su Cheng, with long collaboration with Spin Label. This whole project is done in collaboration with Angela Gronenborn, and is supported by NSF, BSF, US, Israel, and Yang Ping. We collaborate on the Tritil, and Alexander and Leonid helped us with some of the measurement that had to be done with very low temperatures. And with this, I think I'm going to end. And I'm really happy that I could go through the whole 50 minutes without a siren of a missile attack.
Thank you, Daniela. It was a wonderful talk. I mean, as always. And yes, it was great that we could go through the talk without this. That's great. Now we come to my most favorite part, and that is obviously the question session. So if you have a question, either raise your hand or just speak up, and I'm sure Daniela is excited to answer your question. Now, while we're waiting for people to gather a question and speak up, I'm just going to ask very quickly, Daniela, when you do the fluorine ENDOR, like now you showed like, I'm not sure if you showed like, examples where we have actually multiple fluorines on one side. So for example, if I would have, let's say, a methyl group where all three protons are substituted with fluorine, and then you would need to worry about, I guess, fluorine methyl tunneling, I guess.
Yeah, I did show you. There were examples. Examples were 3, and there is some, I think, calculations that were done by Nick Cox and maybe I think by Marina, that if it is, I think, longer than 10 angstrom, Marina can correct me, then the 3 of them can be considered as similar.
But wouldn't we need to worry that they're because of the methyl rotation or the methyl tunneling, that wouldn't that?
I think the methyl tunneling is mostly for protons.
Okay, well, fluorine doesn't matter then. Okay, sounds good.
So in principle they have slightly different distances, but I think there's dipolar interactions.
Right.
So they can cause broadening.
Right, that's what I would have thought.
Yeah, but we consider them as one, like 3 fluorine at 1 distance.
Okay, sounds good. And they all clump together. Unless, yes, it makes sense, right? Because if they're closer together, then yes, it would matter. But if they're farther than 10 angstrom, then the distance would be so similar.
The difference is small, and it's within the.
Yeah. It wouldn't matter then, right? Good, got you. All right, cool. Anybody else have a question? Okay, so
can I speak up?
Oh yeah, please go
because
ahead.
I put my hand up, but apparently you didn't see. I guess there are many people here. Actually, I can have 2 things maybe. So first, my question was about the gadolinium outer transitions. So if you use the central transition, you had as an advantage that you don't have orientation selection in many cases, at least.
That's what we thought at the beginning. So most labels, they were labeled that we use, the ZFS is relatively small and the central transition does not have orientation selection. But when we looked at this cyclodextrin, the zero field splitting was larger and the central transition showed us very, very nice orientation selection.
Oh, okay. But and if you use the outer transitions, you always have orientation
then there is,
guess,
then there
right?
is, yeah.
Okay. And if you use the central transition, how can you be certain that, I mean, the outer transitions lie below the central transition, right? In part. So can you be certain that they don't disturb your measurement?
Yes, because usually I can see them. So if I go to, I didn't bring it here, but okay. So you see, if first, if I work with, okay, so you see here you have maybe 10%, okay, which is hard to see, and then you would see them here. Yeah, so they show up here, right? So you, in principle, you can identify them.
Right, but you had this cyclodextrin example where you had like 10% of another conformation, and then maybe it's hard to distinguish, right?
Yeah, but then I know exactly where they should be.
Okay.
If I know one, I know if, let's see here. Yeah, if you see, all these are related to 1A. I didn't have time to show you, but when we measure in different places, you see these peaks, and they really, you know, all these are, see, this is 1A, this is 1A.
Okay.
So you know where to find them.
Okay. Yeah, thanks. So
Okay.
I can comment on the trifluoromethyl rotation, but only if I don't want to take
comment,
question
because
time
maybe
away.
maybe I gave a wrong
So
answer.
so if I may, so we also measured one trifluoromethyl example, and in our case it was that it was rotating at 50 Kelvin. It depends on where it is sitting, and it was attached to a phenyl group, so similar to the phenylaniline example from your talk, Daniela. And I think at your measurement temperature of 10 Kelvin, it should be non-rotating, so it should stand still. This at least what also came out of our calculations and from our experiment.
Okay, thank you.
All right, great. Thank you, Andreas. Graham, I think you had a question.
Graham?
Can you hear me?
No, not very well.
We see your name, we see your hand, but we don't hear you.
He's Graham, if you want, you can also type in the messenger board your question if you can get your microphone to work.
Good idea.
Yeah, I think he's typing. Hey, since Graham is typing, Daniela, one more question. If you, I mean, it's kind of interesting. I like the idea what you're having with auto transitions to look at it, and then also where you can actually determine based on your dextrin example, where you can actually determine the orientation. Now, if let's say, for example, that would be actually very cool because now one can utilize that method. Let's say in a protein, we have, let's say, 2 fluorine labeling sites, but they are obviously apart by the same distance. But now we can use the orientation, what you have shown in the dextrin example, to still identify where the location of those fluorines is. Is that correct to assume?
I think so. But what we are now trying to do, you know, usually the zero field splitting of gadolinium is, you see, when you do orientation selection in nitroxide and you know very well where your G tensor is, then you can get really very important information from the orientation selection. If your labels are not just wobbling too much, right? And there are nice examples of I don't know, of DNA, RNA, and Marina did, and Thomas Prisner did, etc. So this is really, really nice. But in principle, you could do the same thing with the ZFS of gadolinium. There are 2 problems. So often it's too distributed, but not always. And the other is you don't really know where it is. Right? In copper, you also, usually you know very well. So the question is where it is. And the calculations of ZFS with DFT are not that great. So in principle, you can make molecules that you know the gadolinium distance and orientation.
Yeah.
And then from this, you can determine the orientation of the gadolinium ZFS.
Okay, I see.
And then this should be benchmark for DFT.
Okay, I see. Okay, so then you would use that experiment to calculate
But I
calibrate
think
DFT. Got you.
yeah, but I don't know. Maybe we didn't try. Maybe if you have enough resolution, you can see two that have different orientations. Yes, but then you should have a gadolinium spin label that has a large enough ZFS.
Okay.
If you take the ones that but then yeah, or you go to work. So then you cannot do it on the central transition, but you can do it on the other transitions.
Right.
But I think, yeah.
Right. If you have no resolution, maybe. Got you. Okay. Graham. Oh, I don't know. Graham didn't finish his question yet. Hi Daniela. If you're using the outer transition, is the optimal temperature as low as possible as opposed to 10 Kelvin? Oh, so I think Graham is asking.
That's why we went all the way to Alexander, and he could measure at 2.2. Now we can measure at 3 point something. So going such low temperature is not so easy.
Right. How do you go low? You're using a cryostat, or how do you go that low?
We use a cryo-free.
Yeah.
That in principle should go lower, but when you put the ENDOR in there, it warms it up. Alexander, he's here. Alexander, can you explain why your system goes so low?
No, I cannot explain what the difference between ours and yours is. We have a variable temperature insert also, not a helium cryostat. And this is a very sophisticated balance between the pressure which you have in your cryostat and the temperature. And this is something which needs to be tried out. And yeah, that's so it's not liquid helium, it's a VTI. This is important.
Yeah, we improved it to 3. But I think 3 is enough. Actually, if you can also do it at 5 and 6. Just the analysis is a little bit more complicated because you have more transition. But usually, if you know your A from 1, you can.
Then you don't need to care anymore because you just fix it, done deal.
Yeah, right.
So in theory, you would just need to do one measurement, then you know your A, and the rest is, well, it doesn't matter if high or low.
Yeah, but if I cannot resolve it in my central transition, I don't know my A.
Yeah.
So I need to go outside.
Right. All right. Frederick Mentink, he is asking, Daniela, do you think higher field would help? Larger LAMO for 19F, but also larger population in the -7.5 or -5.5.
I think it will help. I think there is another thing that I didn't talk about. In principle, you have the fluorine signal pretty close to the protons. So for instance, at W band, they are separated well enough. At Q band, you can do all these measurements at Q band with gadolinium also. But at Q band, they are closer. So sometimes there was a nice example, I think that people did. So they measured only the protons and then with the fluorine and then subtracted. So I guess if you go to higher fields, it will be much more resolved, more orientation selection. I don't know about this, and Marina can tell you 263. How easy is that there's publication, and also you resolve in principle, you can resolve very nicely the fluorine chemical shift and isotropy, and this has also some information that you can use to determine orientation relative to something.
All right, Marina, you wanna?
Yeah.
You can
So
answer.
first of all, hi Daniela, how nice to see you. Thank you for the beautiful talk. Yeah, maybe a comment. So the CSA at even higher frequency could become problem in the line width. So
Yeah.
maybe W band. So we haven't done a systematic study, but if we're going to longer distances, then you have to consider CSA, and it broadens.
Yeah, right,
So,
right. And
Yeah.
there are cases that we see it even in W band.
yeah. So so I mean, I would have several questions, but and maybe we will talk separately. But I thought what is intriguing, and maybe you can repeat it. So the comparison between the labels is very interesting. It was striking to see how the treacle is worse than the other. You pointed that out, but so we also work with Elena. I think Elena is also here, and we looked at the treacle fluorine distances, and somehow what makes treacle so much worse? Is that just the tether? That's longer? Is that because everything?
I think depends on depends on the label that you take. So here, you see, if you have this one,
Yeah,
the T2 is horrible.
yeah,
But if you get rid of the methyl, then it's much better. Yeah, so you see that with this one was terrible. Now here the T2 is not so bad. But look, the lengths, you add, it's the length of the tether. It's a huge, look, from here.
yeah. But
from here to here. So if you only measure 20, and from this one label, unless the labels go like this, but if then you lose a lot on the tether.
That's clear.
So
maybe Elena can comment on that. Would it be possible to make this tether shorter than this?
Yeah, so if you can just stick it to...
Yes, one can make it shorter, and also I was wondering about electron spin relaxation, because in our case it was longer, and probably that is exactly depends on the system where you are working. So
you see, if you replace this already made a big difference. So this will give you the longer, and then I think that the main issue is the tether. But if you can just stick it here, it in my opinion, I think it depends what you use it. It's too big of a label.
How about if you deuterate the whole label?
That was exactly the question which we calculate and try to show how big it is and from which distances one can play with this.
But I think
as better. Also, I like very much papers by Marina where she use it for chemical shift for. She extract information from nitroxide, and that was really very, very impressive. Not only just distance, but also a lot of structural information.
Yeah. So it's nice.
This I like very much, because we also, when we start this work with treacle, we thought, oh, very narrow line, very nice. We can just not measure anything. And then when I see paper by Marina, that they also use it for extracting structural information, I really was amazed. A little bit long experiment, but still then much more information. Then it's nice.
Yeah, so
but
it's
very
good,
much,
it's
a very nice talk. So many years we can't see each other due to so many bad things which has happened in our world.
Yeah.
So it's so nice.
I think that it's nice that you have 4 or 5 types of labels, and you choose what is. We have now an example that the only one that will work is a copper. So we use copper.
Yeah, I agree. Copper is
but think about 20 years ago, you only had nitroxide. So now you can choose for this or this, depends on the application, what you want to do. So it's... I did not show, but we also tried to predict the distances. using MMM and MTSL wizard. And for Tritil, it was the most difficult,
Wow.
the prediction for the system we tried.
Because it's all this complicate how to calculate, and it's like a helicopter has this all rotation.
Yeah. So this I didn't have
I
time to
agree.
show, but so
Better to use the simplest. This I agree.
Yeah.
Okay.
How about if you deuterate the methyls? That should probably do something too, right? For the triddle label, what before you have.
I'm sure that this will help.
Yeah,
Relaxation become much longer for
because the deuterants are going to
Yeah,
shield everything.
also
Yeah,
60.
because Tritil has such a narrow line that we thought that it will be wow.
Hmm.
Too large.
All right. That being said, Stefan is asking, great work. Do you have any thoughts on how sensitivity could be increased further? How you can enhance sensitivity?
Okay, I can tell you what we are trying to do, but so far not so great. We have to improve the ENDOR setup. To use to have more efficient ENDOR efficiency. This is one thing. I think for gadolinium, you just do if you can have very, very small ZFS. So then you will have much stronger EPR. Because for deer, very, very small ZFS, you get into trouble. But for ENDOR, not. So if you can have and there are these new labels that I think Janet published with very narrow line, very small ZFS. So this will increase. What else? Beside instrumentation. If you know how to, the problem is, if you want to increase your TM, you lower the temperature, but then you lose your central transition. So lowering the temperature is not.
It's not the solution. Frederick Mentnik is asking, what is the issue with the ENDOR setup? The radio frequency impulses are too long or not homogeneous? So pretty much he's asking, what are you planning to improve on your instrument?
I think that the efficiency of the RF, we put a lot of power and the B2 is not, in principle, what you really want to do, and I think Marina showed it, you want to do time domain ENDOR. And these are small splittings. So in principle, you can cover the whole ENDOR spectrum if you have a good enough B2. But we don't.
Is that why you wanted to do echoes?
No, we did the echoes to measure the T2, yeah. But initially we wanted to do time domain ENDOR and get rid of broadening, but we saw that it was so we've been trying to do all kinds of things to improve the probe itself and have better V2, but so far our system has been the most robust one, and if you can get a pulse that is very short, you can do this time domain ENDOR, and because you have splittings of kilohertzes, it's not the megahertzes.
Yeah, so you have I
But
guess
I
you have
know that Marina, you can do it, right?
Yes, I think we can do, but we use nitrox, so I'm a little bit careful in comparing with your system. I still don't know if there is. So when you go into the time domain ENDOR regime, what?
It just matters
the gadolinium.
you want to cover the full ENDOR spectrum. That's it.
Yeah, that's great. But this is actually, if you have just a couple of kilohertz coupling, is not.
Yeah. So if I could have an RF pulse of, I don't know, let's say 5 microseconds and not 50 and do a time domain ENDOR, then I don't have the broadening, the power broadening.
Ah. Mm. Okay, so yeah.
But I think that even with this setup, there's lots of stuff we can do.
Of course.
Daniel Klose is asking, nice talk, thank you. There are also gadolinium complexes known with positive ZFS, while all labels known so far have negative ZFS. Wouldn't a label with positive ZFS and hence very low temperature sharp central transition be expected to perform better? So in other words, weighting the benefits of negative ZSF and positive ZFS.
I don't think that it matters if it's positive or negative. I think what matters is first you don't want to have a very large distribution, and then you want it to be the smallest as possible. So you will get a very narrow line and very strong line. If it's negative or I don't think it matters if it's positive or negative. And many of the spin labels that have this large distribution, at least according to Arnold Weizmann analysis, they have both positive and negative. So I don't know. I don't think it matters, but maybe it affects the T1. I don't know how it affects the T1.
Anyways, all right. I think does anybody else have still a question? Otherwise,
how about Graham's typing? He got stuck.
no, I think you answered his question already.
Okay.
His question was just like how low should one go, and I think as low as possible is a good answer. Oh, then Daniel got back. But Daniel, if you want, you can speak up. That's fine. Well, Daniel said, well, positive ZFS would have very sharp line and very low temperature for efficient microwave excitation. So what he's saying is, well, if we would use a positive ZFS, you would expect for very low temperatures that there would be a better microwave excitation. Therefore, you get better sensitivity. That's what he's saying, I think.
I think it's just a mirror image of the spectrum. But I don't know. I need to check. I know the spectrum, usually if you have a positive or negative one, it's just a mirror image. But I don't know. I need to think of it.
Yeah.
Intuitively, I think it doesn't matter, but maybe I'm wrong.
Right. So Daniel says, thanks. Good. Stefan kind of says, okay, well, one could do an easy spin simulation, might figure that out rather rapidly. But either way, I think let's see.
Okay, Stefan, do a simulation and let me know.
There you go. All right. Well, with that being said, I think that kind of ceased most questions. Oh, never mind. Graham came back. Graham's question was are you limited by the T1 at low temperatures? Like the reason why you go to the lowest temperatures, is it because you're limited by the T1? Is that the reason why the lowest temperature is the best? That's what
no, I go to lowest temperature because it depends for what. If I just want to do standard ENDOR on the central transition, I'm going to go to 10K. I'm not going to go too low. I usually go to the lowest temperature because I want to resolve the lowest EPR transition that gives me the highest resolution in terms of the splitting. Okay, so it has nothing to do. So this usually, if you go lower, then your T1 becomes longer, and this you pay by your sensitivity, right? Because you cannot accumulate that fast. When we looked at the T, at what affects the phase relaxation of the nucleus, then the first thing you do, you look at the temperature dependence, right? And then when you see a temperature dependence, you say, okay, this must come from T1. And that's how we ended up with this dependence on T1 that is one of the limits of, at least according to the paper of John Morton. And we were surprised that it fitted so much. So according to this, you go lower, then your TM will become longer. But if you go very, very low, you're not already at the central transition. So it's so it really depends what you want to do.
I think Marina wanted to speak up. Go ahead.
Okay, I think I really would like now to ask also this question. It's about the resolution limit and your measurement that you estimate basically would be something like a few kilohertz based on that.
No, it's not estimated, it's measured.
No, I know. Yeah. Well, wait. So you measured it too. So my question is there anything else between T2 homogeneous and what you could measure? So I'm just worrying about what if there are maybe other pathway of relaxation that could be in between.
So okay, what we measure
because you measure T2 homogeneous, but the line, so in the line with
no. So I measured the homogeneous.
Could be,
yeah, no. So the line width has lots of things, and I think that the most okay.
Okay, they are the broadening mechanism, but on relaxation pathway, could there be maybe more than that? I'm just I'm puzzled by this question. Yeah, if that is really.
This is what we measured. Now, for protons, for instance, we did some measurements on protons. Protons, it's not, because we have here one fluorine, right? So now for protons, we saw that for protons, it really depends on the hyperfine. And if you have very small couplings for protons, you get nuclear spin diffusion. And this for proton was very clear. We added a little bit of H2O, booms. We saw a change, but only for the very small couplings. For the larger one, not.
Yeah.
So, so for the fluorine, what we found so far for this seemed to be T1, but you see, it's not exactly on the line. But we did not find any correlation with the hyperfine. In principle, maybe they should.
Yeah.
But
yeah.
So there's another problem that we measured the echo decay, but because the SNR is not so great, we could not go to very long times.
Mm-hmm.
So I guess if you could go to longer times, you get a better curve, and maybe you can see that you have, something more complicated.
Okay.
But in any case, what we measured is much smaller than distance distribution dipolar couplings.
Yeah.
So you have to worry first about distance distribution and then about lengthening.
Of course. And maybe Fred can comment also on that.
What?
Yeah, I have a like the equation of that you report, the one from Morton, is only valid as long as the inverse of a T1 is smaller than the splitting you have in between your two peaks.
Right. So we are in the So normally, if you take the NMR, the equations that NMR people use for paramagnetic relaxation, they don't fit at all, because you are in what we call the slow exchange limit. That you right.
But then it should not be hyperfine dependent. Beyond this point, it should not be hyperfine dependent.
Yeah. We did not see it because we measured the distances that we had were like 10 and 11, or, although the difference in the coupling is large, in the distance it's small.
Okay, thank you.
Okay, guys.
All right.
So
Great.
it was nice to see you. I'm sorry I could not see everyone on the screen.
Well, don't worry. We appreciate your talk. I think it was a great talk. And as you can see, a lot of people have a lot of questions, which is an indicator for a good talk, I guess. With that being said, most
Okay.
importantly, thank you, Daniela, for giving the talk. And thank you for everybody to attend this great presentation. I'm looking forward to see everybody again. So thank you, Daniela.
Okay. I didn't see the chat. What was in the chat? Oh, there are some questions
Oh,
here.
those are just the question I asked.
Ah,
So,
okay.
boy.
Take
Thank
care.
you.
Bye-bye.
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 Bye.
Bye bye.
Bye.
Bye.